Alright, in this last lecture on respiratory system physiology, we're going to put together what we've covered in the previous lectures, and we're going to talk about control of respiration. Why do we breathe as fast as we breathe or as slow as we breathe, and how does the respiratory system adjust to different conditions that we have to deal with? Let's first talk about, in general, the mechanisms of control of respiration. Of course, homeostatic mechanisms are going to intervene anytime we need to meet the needs of the body in terms of cellular gas exchange. So that would mean increasing the concentrations of oxygen in the body, getting to the tissues, or decreasing the concentration of carbon dioxide that have to be expelled from the body. In a similar way to what we've seen before, control occurs at two levels. Number one, we have local regulation. If you remember when we talked about the cardiovascular system, this implies that regulation is at the level of the tissues without having to involve the nervous system. And we're going to talk about each of these in turn. The second mechanism is respiratory centers of the brain. In other words, we're going to begin involving the nervous system. And of course, the respiratory centers can override local regulation in some cases. Now, I want to talk about this concept called ventilation, perfusion, coupling, or matching. We use the term V to indicate ventilation, and we use the term Q to indicate perfusion. Ventilation simply means the flow of air past respiratory surfaces. Perfusion implies the flow of blood. And when we talk about a VQ ratio, we're talking about the ratio of air to blood in any given portion of the lung. Usually the lung perfusion, that is the flow of blood through the lungs, is on the order of about 5.5 liters per minute. And if you remember, this is about the same as the blood supply. So basically we're saying that the entire blood supply moves past the lung every minute. Alveolar ventilation is about 4.2 liters or so every minute. So this is how much air we're moving in and out of the respiratory surfaces every minute. And what we want to do is we want to match these two things. In other words, we want to match the amount of air that we're bringing in to the amount of blood that we have in an area of the body. When we talk about local control, we're talking about this process regulating both the efficiency of oxygen pickup in the lungs as well as the rate of oxygen delivery in each tissue. So let's discuss those in a little bit more detail. Remember, this is the ventilation perfusion ratio that we looked at in the last slide. Ventilation being the airflow, perfusion being the blood flow. What we'd like to do is to match these two as closely as possible. So if we look at an illustration of this, here we have an alveolus in the lung, and around the alveolus we have the alveolar capillaries, and into the alveoli we're bringing in fresh oxygen that needs to get in the blood, and the blood is flowing past the alveoli here. So what we would like to do is to perfectly match the amount of oxygen that we could bring in the blood with the amount of blood flowing past that alveolus. Now sometimes what happens is the oxygen, for some reason, is not making it down to the alveoli in a particular lobule of the lung. And if you think about this, it would make no sense to send a great deal of blood to those lung lobules that don't have a very high amount of oxygen. There'd be no reason to do that because the blood wouldn't be oxygenated. Instead, what we'd like to do is shunt the blood away from those lobules that have a low amount of oxygen to the lobules that have a high PO2. And so what happens is those lobules in the lung that are not ventilating well, so what happens is the alveolar arterioles constrict when the local PO2 is low. Now I want you to think about something we talked about before in the tissues when we said that the tissues were working hard and the local PO2 was low. The arterioles, if you remember, dilated to bring more blood to those hard-working cells. I want you to notice that this process that we have going on in the lung is the opposite of what we have in the tissues, where we actually dilate arterioles to bring more blood when we have poor oxygen in an area. In contrast, in the lungs, when we have a lobule that's ventilating poorly, we do not want to send a lot of blood there. Hope that's making sense to you. The alveolar ventilation can also be governed by the size of the respiratory passages leading to particular lobules in the lung. So in other words, here we can affect airflow as well, or ventilation, which we characterized as V before. When we have high levels of PCO2, which is called hypercapnia, this causes bronchodilation. That should make sense to you because if we have high PCO2, that means we have to get rid of more CO2, and we'd like a bigger area to send that CO2 out of the body, so we bronchodilate and we can move more CO2 out with every breath. On the other hand, when PCO2 is low, which we would call hypocapnia, This causes a bronchoconstriction because there isn't as much CO2 to move out. Okay, so what this basically does is now this directs airflow to lobules that have a higher PCO2. On the other hand, what we do with the blood is we direct the blood flow to lobules that have a high PO2. This is what we characterize as basically the ventilation perfusion coupling in the lungs, and this is how we govern the oxygen pickup efficiency within the lungs. Now, as we said before, we also have to govern the rate of oxygen delivery in each tissue, and we're talking about changes in partial pressure of oxygen and CO2 local in the tissues. And remember that local vasodilation in the tissues will occur when these tissues are working harder and the local PO2 is low. Okay, so these are kind of opposite processes in terms of how the lung arterioles work and how the tissue arterioles work and how they respond to different concentrations of O2. Now, the diameter of the bronchioles can be controlled. If you remember when we talked about respiratory anatomy, we discussed the bronchioles and said that these were structures that had no cartilage in them. All these have is smooth muscle. So these are the air passageways that can really bronchoconstrict, which means to reduce the lumen or the opening, or bronchodilate to increase the size of the opening. And the muscles around these bronchioles respond to a few different things. Bronchodilation is accomplished by things like epinephrine, sympathetic stimulation, the fight or flight response. And if you remember, we mentioned when we talked about the lymphatic system, anaphylaxis. And we said one of the antidotes basically for an anaphylactic reaction is an injection of epinephrine. One of the reasons for that was that we would dilate air passageways. So this would allow the person to get more air. Now, bronchoconstriction can take place from a number of things. Parasympathetic stimulation, so rest and digest type activation of the autonomic nervous system. Histamine, part of an allergic reaction, that will cause bronchoconstriction. And the epinephrine that we could inject in an anaphylactic reaction or an allergic reaction can counteract the effects of the histamine. Cold air can also cause bronchoconstriction. This has to be kept in mind when you're dealing with elderly patients. We talked about the fact that the nose and the respiratory passages, the conducting portion of the respiratory tree, one of their functions is to warm the air on its way down to the lungs. So with elderly patients, especially in very cold weather, it's important for them to breathe through the nose rather than the mouth so that the air has more time to stay in contact with the warm mucous membranes and be warm before it makes it down to the bronchi so that we don't have any kind of constriction of these air passageways. There are some chemical irritants as well that can close or constrict the bronchioles, and these would of course reduce airflow to the lungs. Now, the surface tension of the alveoli and the distal bronchioles is another factor in resistance to airflow. If we have more fluid collect in portions of the alveoli, as we talked about, or we have more mucus collect in the distal bronchioles, all of these things increase surface tension. They basically will squeeze shut these passageways and the alveoli and make it more difficult to breathe because they'll increase the resistance to airflow. Now let's talk a little bit about the neural control of respiration now that we covered the local control a little bit. If you look at this diagram, you'll see a schematic diagram of some of the major areas that affect respiration in the central nervous system. Remember from Anatomy and Physiology 1, here we would have the midbrain, here we would have the pons, and finally down here we would have the medulla. Notice that toward the rear of the pons we have something called a pneumotaxic area. There's also something down here called an apneustic area that I'll show you in a little while. These two are basically regulatory centers that will affect breathing depending on different conditions in the body. However, the two that you see in the medulla down here, these are known as part of the medullary rhythmicity center. And there are two of these. There's one in the back that's called the dorsal respiratory group. We abbreviate this DRG. And there's one in the front that's called the ventral respiratory group. We abbreviate this VRG. Now, these are the two respiratory centers that govern eupneic breathing. And when eupneic breathing changes due to different conditions in the body, the DRG, as we'll see in a second, can affect the ventral respiratory group. So let's take a closer look at these. The ventral respiratory group, as you see in this illustration, is the one that is thought to normally generate the rhythm of breathing. In other words, this is the one that will fire periodically, cause the diaphragm to contract, move downward, cause the external intercostal muscles to contract so that the rib cage expands. Remember, we talked about the fact that between those two things, the thorax expands. We take in about 500 milliliters of air normally. That was called the normal tidal volume. On the other hand, the dorsal respiratory group is thought to integrate input from different things in the body and send that information to the ventral respiratory group to modify how it works. So it's believed now that the ventral respiratory group is the one that controls normal rhythmic breathing, while the dorsal respiratory group is the one that can modify that normal rhythm to suit different circumstances. Together we can say that the VRG and the DRG basically regulate the rate and depth of breathing in different circumstances. And you can see this what we refer to as the pontine respiratory center. This is the one that we talked about before as the pneumotaxic area. Okay, so let's look at the connections between these things. And what we really want to start talking about now is what happens when conditions change in the body. How does the respiratory system accommodate those changing conditions to breathe faster, for example, to breathe slower, breathe deeper, breathe more shallow? Here you see the anatomy that we just talked about. And here you can see another illustration of the dorsal respiratory group, the DRG, the ventral respiratory group. Remember, both of these are in the medulla. They're part of the medullary rhythmicity center. And here we see the two centers in the pons, what we would call the pontine respiratory centers. This is the pneumotaxic area, as well as the apneustic area here. These take input, as you see, from a number of different things. Okay, so we have some higher centers in the brain that can affect breathing through the pneumotaxic center. The apneustic center can be affected by stretch receptors in the lungs. And importantly, we wouldn't want the lungs to stretch too much. And basically what happens is we start to inhibit stretch of the lungs by inhibiting inhalation when the lungs get to a certain capacity. And the apneustic center is part of that inhibition. We're going to talk about that reflex in a little while. Now, as we said, the VRG is thought to control normal rhythmic breathing. The DRG modifies the output by the VRG, depending on local circumstances. What we want to talk about here is what are those local circumstances that might change the rhythm and depth of breathing. Basically, what we're talking about here is responding to chemical composition of the CSF. Notice these things in here are called CSF chemoreceptors. As we discussed in the sensory lectures of the nervous system, chemoreceptors respond to different concentrations of chemicals. So these are receptors that are built into these areas here that respond to changes in chemicals. Specifically, what they're going to respond to is levels of acid. So what we can say about these central chemoreceptors is that they respond to pCO2 concentrations and pH of the cerebrospinal fluid. Now this is the reason why we call these central chemoreceptors, because these are located within the CNS. Importantly, they respond to pCO2 and pH changes. Well, think back to the equation that we talked about before. Let's look at how this is related. We said before that if we increase something on this side of the equation, what we would do is drive the equation this way and ultimately produce more of what's ever on the other side. So let's talk about what happens when we increase CO2 in the body. Now, one way we could do that is by not getting rid of CO2, for example, by holding our breath. What that's going to do is, since the cells are still putting out CO2, even though we're holding our breath, they're still metabolizing, and they're putting CO2 into the blood, but we're not getting rid of it, that's going to cause levels of CO2 in the CSF to start going up. How is that going to affect acid? Well, as we said, when we increase something on this side of the equation, we're going to drive it this way. Ultimately, what we're going to do is increase concentrations of acid or hydrogen ion in the CSF as well. And so here the chemoreceptors are going to respond to this increased hydrogen ion concentration, and what they're going to do is send signals out to the diaphragm and the external intercostal muscles, and the other muscles to accommodate breathing. They're going to send signals to make us breathe more rapidly. So anytime we have an increased concentration of acid in the CSF, we stimulate more rapid breathing, deeper breathing. What this is going to do is get rid of more CO2. So something that's important to keep in mind, especially in the lab that we're going to do on this, is the faster we breathe, the more CO2 we blow out. And that's exactly what these central chemoreceptors do. They adjust our breathing rate so that we maintain approximately normal levels or homeostatic levels of CO2 in the body. We don't want to let this get too high. We don't want to let this get too low. Now, we also have some chemoreceptors located outside the central nervous system. For that reason, we refer to these as peripheral chemoreceptors. These also respond to pH and CO2. However, these also have the capability of responding to oxygen concentration in the body. Now, I want you to take a look at the statement on the bottom of this slide, and it's very, very important that you remember this. In normal physiology, with people without any kind of respiratory pathology, CO2 is the most powerful respiratory stimulant. Increasing levels of CO2 are what are going to stimulate breathing. I don't know if anybody's ever seen this. You know, when children threaten to hold their breath and kill themselves, well, they really can't. The reason for that is even if they held their breath long enough to pass out, CO2 levels would keep rising because they're still alive. The cells are metabolizing, making CO2, and that would basically cause them to continue breathing even if they held their breath. So CO2, once it reaches a certain level in the body, is actually going to stimulate the respiratory centers to make us breathe. Okay, we're going to talk about that a little bit further in the laboratory. The point that we want to make here is that we have peripheral chemoreceptors that can not only respond to concentrations of acid, but can also respond to concentrations of oxygen via PO2. And these are located, as you see in the diagram on the right, in the carotid bodies as well as the aortic bodies. So here and in here as well. We talked, remember, about the locations of baroreceptors in these areas when we talked about regulation of blood pressure. So located in the same place as the baroreceptors, we also have chemoreceptors that are very important in governing the breathing rate. We have, as we said before, motor impulses that travel from the medullary rhythmicity center, and these will eventually, ultimately, affect the diaphragm, as well as the external intercostal muscles, and even some of the other assister muscles that we talked about if, for example, we want to take a deep breath in or we want to expel more air out. So these motor impulses will travel down to whatever muscles we need to accommodate breathing. The second thing is, notice that as the contraction of these muscles continues, the lungs expand further. If, for example, we stimulate the diaphragm more than usual, or stimulate the external intercostal muscles more than usual, or the sternocleidomastoid muscles, or the pectoralis minor muscles, remember that all of these muscles serve to help us take in air. And when we have contraction of these muscles, the lungs continue to expand. But just like a balloon that can stretch too much and eventually burst, what can happen is if the lungs stretch too much, we can actually have tissue damage occur. So we have to limit the expansion in the lungs, and the way that's done is with an inhibitory impulse that's known as a Herring-Brewer reflex. This has to do with what we looked at before, and that is the apneustic center receiving inhibitory impulses from the sensory nerves inside the lungs. In other words, these sensory nerves are tied into the apneustic center. This is going to inhibit further inflation of the lungs so that the tissue of the lungs is not damaged. And once again, just as a reminder, CO2 is the most powerful respiratory stimulant in people with normal physiology. And let me just mention as an aside that in people that have chronic hypoxia, that is low oxygen in the body, from conditions like COPD, like emphysema, those kind of things can actually cause a resetting of the chemoreceptors. And in people with respiratory pathology, sometimes oxygen becomes the primary governor of respiration. And that can be dangerous because if we see somebody that's having a problem breathing, and if they've had a chronic respiratory condition for a long time and we slap oxygen on them, what can happen is it can actually cause them to stop breathing because we've had a switch between the governors of respiration. Now, instead of CO2 being the main control for respiration, oxygen is. So just as a summary, I want to just review these things very quickly. Remember that control of respiration is accomplished by two things: local regulation and also nervous system regulation. When we talked about local regulation, very importantly, we talked about that important VQ ratio, so ventilation, perfusion, matching, or coupling. And what we said, remember, is that if we have decreased alveolar ventilation, for example, in a lobule of the lung, we would want to decrease blood flow to those alveoli. Conversely, if we had an increased alveolar ventilation, we'd want to increase the amount of blood that we send to those alveoli so there's more hemoglobin available to pick up the increased oxygen. We also talked about bronchodilation and bronchoconstriction. If we have an increased alveolar CO2, we have bronchodilation to rid our bodies of that CO2. If we have decreased alveolar CO2, bronchoconstriction will take place because we don't need to get rid of as much CO2. We talked about nervous system control in terms of the ventral respiratory group and the dorsal respiratory group. Remember, this is the one that normally governs rhythmic breathing. So eupneic breathing is typically governed by the VRG, or ventral respiratory group. And then the dorsal respiratory group can send input to the ventral respiratory group if conditions change in the body and we need to change either the rate or depth of breathing. We also said, very importantly, that CO2 is the most powerful respiratory stimulant. Normally, our impulse to breathe is governed by levels of CO2, so PCO2. This equation is very, very important. You need to know this equation cold. And this is, as we said, CO2 in water are going to be converted into this, which is, carbonic acid. And the carbonic acid is going to dissociate into hydrogen ion and bicarbonate. Importantly, we mentioned that if we increase CO2 in the body, we also increase hydrogen ion concentration. And this is primarily what's detected by the receptors that you see below, and we have two sets of those. We have peripheral chemoreceptors that are located outside the central nervous system in the aortic and carotid bodies. Importantly, these can also be sensitive to oxygen, whereas the central chemoreceptors mainly rely on CO2 and acid. Okay, so that finishes up our respiratory lectures. I will see you next time when we'll begin the digestive system with Lecture 9.